Hi everyone, it's Dr. LeBlanc with Unit 15A, Viral Genetics, the viral reproductive cycles. Our learning goals are to understand the structure and function of virus particles, to understand how DNA virus particles reproduce and how RNA viruses reproduce, to understand the different types of infections mounted by prokaryotic viruses or bacteriophage and eukaryotic viruses to understand lambdaphage as a model system. Here's a catalog of some of the various types of viruses that you can find here on planet Earth. The first one is tobacco mosaic virus, and it has a very interesting and different structure to it. It has an RNA genome that is basically coded by a single type of protein, and it can be quite long. Adenovirus is the virus that often causes the common cold. It has an envelope, it's a non-enveloped virus with a polyhedral capsid and these protein fibers with these little knobbies on the end. And inside is DNA. Influenza virus is an enveloped virus, which means that it has a membrane derived from its host cell around it, and then a protein capsid on the inside, and an RNA virus, an RNA genome as well. And here's our old friend T4 bacteriophage with its spaceships like function structure with its DNA in the middle and its the shaft and tail fibers that contact the bacteria cell. So the reproductive cycles of all these viruses have certain features in common. There are 6 steps that you can find during this reproductive cycle. First is attachment to the host cell surface. All viruses have to somehow get their genomes inside the host cell, and the first step is to attach to the cell. The second is entry into the cell, and there are various mechanisms by which that occurs. Sometimes the viral genome can integrate into the host genome. Then all viruses have to synthesize all their components. There are multiple copies of their own genome as well as all the proteins that make up the capsid, and then a few extra proteins too, like their own DNA polymerase or reverse transcriptase. And finally, the proteins and the nucleic acids all get assembled into more particles that can go on to infect, and then somehow the viruses are released from the host cell. Lambdaphage is a very well studied model of bacteriophage, and the first step is its attachment to the bacteria cell and then injection of its DNA genome into the host cell. Now, often lambda will integrate into the host cell, and this process is very well studied, has been very well studied. There's a specific protein called integrase that will mediate this integration of the bacteriophage genome into the host genome. Once it has integrated into the host genome, it is referred to as a prophage. The bacterial cell, if it encounters a change, it's an environment, will start to produce some proteins inside, which will cause the viral genome to excise from its location as an integrated genome, as a prophage. And when it excises, it triggers the viral genome to start to produce all the proteins and other components from its own genome. So it will start to make all the capsid proteins and the tail proteins, as well as more copies of itself. They get assembled, and then they will be released from the bacteria cell to go on and infect other bacteria. That's the lytic phase. That's the lytic cycle that we just described. But what happens also sometimes is the lysogenic cycle. Lysogeny is the state when a viral genome is integrated into the host genome. When that happens and the bacteria divides and makes more of itself, that viral genome gets replicated along with the host chromosome until there's some environmental trigger which causes its excision, and then it will enter into the lytic cycle. So lambdaphage has been very well studied, and the switch from going from lysogenic to lytic has been also very well studied. HIV is a eukaryotic RNA virus. And if we follow the life cycle of an HIV, we'll find some features that are common to many other eukaryotic viruses as well. So first of all, the virus particle itself is coated with uh, an envelope, and then inside is the capsid, here indicated in green, and inside that is the RNA genome, which in this case also carries a couple of molecules of its own reverse transcriptase. So the first thing, again, during our viral infection is attachment. Some of these, the glycoproteins on the envelope bind to receptors on the cell surface. And then the capsid, the envelope fuses with the membrane of the cell that's being infected. The viral envelope fuses with the host cell membrane, and then when that happens, the capsid is released into the cytoplasm of the cell being infected. Proteases then will break up the capsid and release the RNA and reverse transcriptase. So the first thing that happens is that this RNA genome gets transformed into a DNA genome by reverse transcriptase, and then that DNA genome goes into the nucleus and integrates into the host genome. While it's integrated, it produces more of itself, one long RNA transcript that goes out into the cytoplasm. This RNA transcript has a couple of different options. It can be packaged into new virus particles, but before it does that, you have to make all the protein components of the capsid and the spike glycoproteins that are part of the envelope, and some more reverse transcriptase. And when this is all these proteins have been synthesized and enough RNA, then you'll start to make new virus particles that are shed or budded from the surface of the cell. So HIV doesn't kill its host cell, the individual cells, sometimes, usually, but it can be released and the host cell can remain intact. And that's typical for many other eukaryotic viruses as well. Here's our old friend Bacteriophage T4, 5 different proteins in the tail fibers. It'll be interesting to determine which of these are actually necessary to find receptors on the surface of the bacteria cell. Here's an electron micrograph image of one of our own viruses that we've isolated here at Merrimack College, called Texage, long tail. It has tail fibers right down there. You can see them pretty clearly. When a single virus particle infects a bacteria cell on a lawn of bacteria on a Petri dish, it will cause the lysis of that bacteria. When that bacteria is lysed, it will release more phage into its environment, which will go on and infect the surrounding bacteria, and so on, until you end up with clear spots on a lawn of bacteria. Each of these clear spots contains lots of viral particles in them, and you can go in and harvest bacteriophage from these clear spots. Down here we see an image showing 2 different types of plaque morphology. Clear spots in which all the bacteria here are dead, and lysed open, or cloudy plaques, which have a cloudy center and a clear ring around it. That cloudy center here contains bacteria into which the viral genome has integrated into the host genome. So the middle parts of these cloudy plaques have living bacteria cells with viral prophage integrated into the host genome. So in summary, viruses are biological particles that contain a protein coat and a nucleic acid genome. Viruses can have either DNA or an RNA genome. Viruses with RNA genomes are known as retroviruses. Viruses can infect either prokaryotic or eukaryotic cells. Viruses often have a head or capsid into which the nucleic acid is packaged, a tail, including tail fiber proteins, which interact with the receptor proteins on the surfaces of the host cell. The binding of viral particles to the surface of the host cell, also known as adsorption, is a very specific interaction. During the life cycle of a virus, the particle attaches to the surface of the cell and injects its DNA into the cell. The cell is taken over and starts replicating the viral genome and packaging it into newly produced viral particles. In bacteria, this results in cell lysis. Viral genomes can also integrate into the host genomes where they can remain dormant until an environmental cue triggers its excision and the production of new viral particles. The dormant state is known as lysogeny. Prokaryotic viruses are detected by the formation of plaques, or cleared areas on a lawn of bacteria. Plaques can be clear, indicating lytic infection, or cloudy, indicating a lysogenic infection. Lambda-phage is an example of a temperate phage, a phage that can mount either a lytic or a lysogenic infection.